When we talk about a master blueprint, there's this built-in expectation of action.
Right.
Like,
Yeah, like it's going to do something.
exactly. Like if you have the architectural plans for a skyscraper, you still need the cranes, the steel, and, well, the construction workers actually pouring the concrete.
The blueprint itself just sits on a table.
Right. It doesn't build a thing. But when we step into human biology, we tend to talk about DNA as if it's this, I don't know, this magical tiny architect actively constructing your traits.
Which is a huge misconception.
It really is. And looking through the stack of sources today, which, by the way, covers everything from foundational anatomy texts to Nobel-winning historical papers, it becomes incredibly clear that DNA is really just the static reference material.
It's just the manual.
Yeah, just the manual. So today we're going straight to the actual construction site. We're going to look at the machinery of protein synthesis. Whether you are studying for a biology exam or trying to understand how vaccines work or just, insanely curious about your own cellular machinery, our mission for this deep dive is to bypass all the dense jargon. We want to give you those ultimate aha moments.
And this really is the ultimate biological assembly line. I mean, to understand any of the modern medical applications we're going to get into, we have to look at the two distinct stages of this process.
Right, the two big steps.
Yeah. So first, you have transcription, which takes the static genetic information locked in your DNA and copies it into messenger RNA or mRNA.
Okay.
And then you have translation, where that mRNA physically moves to a ribosome and gets decoded into a functional protein.
I want to spend some time on the mechanics of that first step, transcription, because the source material brings up a really persistent misconception.
That DNA builds things directly.
Exactly. DNA never directly builds anything, and it never actually leaves the nucleus of a eukaryotic cell. I found myself, as I was reading, thinking of the nucleus as this highly restricted reference library.
Oh, that's a good way to look at it.
Right. You have this incredibly valuable master text, your genome, and you absolutely cannot check it out. You can't risk taking it to the messy, hazardous workshop floor of the cell.
Yeah, the vault stays locked. Absolutely. Instead, an enzyme called RNA polymerase accesses that vault.
Like a librarian making a photocopy.
Exactly like that. It binds to a specific sequence just upstream of a target gene, unwinds that localized section of the DNA double helix, and synthesizes a complementary single-stranded copy.
And that copy is the pre-mRNA, right?
You got it. The chemical language is nearly identical to DNA, just substituting a base called uracil in place of thymine, and using a slightly different sugar backbone.
But the sources emphasize that this pre-mRNA copy is heavily edited before it's allowed out of the nucleus.
Right, it doesn't just leave immediately.
Because it's full of these non-coding regions called introns. They get chopped out by this complex called the spliceosome, leaving only the coding regions, the exons, to be glued together.
Yeah, splicing.
And I just found myself wondering why the cell operates this way. I mean, it seems incredibly inefficient to copy all this junk DNA just to immediately cut it out before the mRNA even leaves the nucleus.
It looks totally inefficient until you look at it through the lens of evolution.
Oh, really? How so?
Well, those introns, they act like spacer regions. Because the actual coding regions, the exons, are separated by these huge spaces, it makes it much easier for evolution to shuffle those exons around over millions of years without breaking them.
Oh, so it's a feature, not a bug.
Exactly. It makes the genome modular. You can mix and match functional domains of proteins to create entirely new ones.
Wow, that is incredibly smart.
It really is. So once the spliceosome finishes cutting out those spacers and joining the exons, it adds a protective cap and a poly-A tail. This just prevents the RNA from degrading.
So now we finally have a mature mRNA.
Right. And now it is ready to exit through a nuclear pore into the cytoplasm.
Out into the workshop.
Yeah.
Which brings us to the actual manufacturing phase.
Yeah.
Translation.
The fun part.
Yeah. This happens at the ribosome, which clamps onto the mRNA strand. And the text notes, it's looking for a very specific starting point, the start codon, which is always the sequence AUG.
Right, which codes for the amino acid methionine.
But the part of the mechanism that really fascinated me in the reading was how the raw materials, the actual amino acids, get delivered and assembled.
Yeah.
Because they don't just float into place on their own. We have transfer RNA or tRNA doing this work.
Yeah, the geometry of tRNA is what makes the whole system function. You can visualize a tRNA molecule as a highly specific physical puzzle piece, or like a key.
Okay, key.
On one end of the molecule, it is chemically bonded to one specific amino acid, but on the other end is a 3-base sequence called an anticodon.
So it's literally a physical matching game.
Exactly, a physical matching game. The ribosome exposes the three-letter codon of the mRNA, then the tRNA floats in,
And if it
and
doesn't match?
it gets rejected. But if its anticodon is the exact geometric and chemical complement to that mRNA codon, it locks right into the keyhole.
That is wild.
And once it's locked in, the ribosome catalyzes a peptide bond. It basically severs the amino acid from the tRNA and links it to the growing protein chain.
And then the whole machine just moves forward.
Yep. The ribosome physically ratchets forward exactly one codon, a shift they call translocation, and the next tRNA puzzle piece tries to fit in.
And this ratcheting process continues until the ribosome hits a sequence like UAA or UAG.
A stop codon.
Right, a stop codon, which doesn't have a matching tRNA at all. Instead, it binds a release factor that frees the completed protein chain. So the building is done.
Exactly. It folds up, sometimes with the help of chaperone proteins, and goes off to do its job.
But digging into the code itself, I notice a detail in the historical papers that totally makes sense in hindsight. If I were designing a biological code to pack maximum data into a microscopic cell, I would overlap the code. I would make the last letter of one word the first letter of the next.
Oh, totally. And that was actually the prevailing assumption among early molecular biologists.
Really? They thought it overlapped?
Yeah, they mathematically deduced that the code had to overlap to maximize storage efficiency on the DNA strand. Space is limited, right?
Right. So what changed their minds?
It took some really brilliant experimental work by researchers like Marshall Nirenberg and Har Gobind Khurana. They definitively proved that the code is read in fixed, discrete, non-overlapping triplets.
So just word 1, word 2, word 3.
Exactly. No sharing letters.
But the code also has a ton of redundancy, right? Because out of 64 possible three-letter combinations, there are only 20 amino acids. The sources refer to the code as degenerate, meaning multiple different codons will summon the exact same amino acid.
Yeah, like leucine, for instance.
Right, leucine has 6 different codons. And again, my instinct as an armchair engineer is that 6 codes for one output is a massive waste of space.
It seems like it. But Francis Crick provided the mechanism for why this redundancy exists with his wobble hypothesis, and it's actually a brilliant evolutionary safety net.
A safety net. How does a wobble make it safe?
Well, he demonstrated that while the first 2 bases of the tRNA and anticodon have to match the mRNA codon perfectly, the third position has a bit of spatial flexibility.
It can literally wobble.
Right. It can wobble and still lock in. This means if a random genetic mutation changes that third letter in your DNA, the tRNA often still recognizes the first 2 letters and delivers the correct amino acid anyway.
Oh, wow. So the protein still gets built flawlessly despite a mutation in the blueprint.
Exactly. It's basically a built-in typo corrector.
That is wildly elegant.
Yeah.
But, reading through these historical debates from the mid-20th century, it's funny how obvious this whole DNA to RNA to protein pipeline seems to us now. The early biochemists looked at DNA and completely dismissed it.
They did. They thought it was way too simple to hold the complexity of human life.
Which is crazy to think about now.
But you have to view it from their perspective, They were looking at proteins which are built from this 20-letter amino acid alphabet, folding into these infinitely complex, beautiful 3D structures.
Sure, proteins look like the main event.
Right. And then they looked at DNA, which is just a monotonous repetition of 4 letters. Under the tetranucleotide hypothesis of the time, they assumed DNA was just a dumb structural scaffold.
Just the physical support beams.
Exactly. They firmly believed proteins had to be the actual carriers of hereditary information.
It's the equivalent of opening up a computer hard drive in the 1950s, looking at the ones and zeros, and saying, "This is too boring to possibly run a video game." The monitor has millions of colors, so the monitor itself must hold the instructions.
That is a perfect analogy, and it wasn't until experiments by Avery, McLeod, and McCarty in the 40s, and then Hershey and Chase in 1952, that the consensus actually shifted.
Hershey and Chase, they were the ones with the radioactive viruses. Right?
Yeah. They used radioactive labels on viruses to prove that only the viral DNA, not the viral protein coding, was entering bacteria to pass on genetic instructions.
And once that paradigm shifted and we accepted DNA as the blueprint, the race was on to crack the language. Which brings us back to Nirenberg and also Heinrich Mathai, who achieved this in 1961 with what has to be one of the most elegant experiments I've ever read about.
The poly-U experiment.
Yes. They synthesized an artificial mRNA strand entirely out of uracil bases. just a polyutale.
Right, just UU.
And they just threw that into a cell-free soup of translation machinery, right?
They did. And the ribosome dutifully read those UU codons and spit out a polypeptide chain made entirely of the amino acid phenylalanine.
Just a chain of the exact same amino acid.
Exactly. It was the Rosetta Stone for cellular biology. They definitively proved that UU codes for phenylalanine. And within a few years of that, the entire 64 codon dictionary was mapped out.
Which perfectly sets the stage for Francis Crick stepping up in 1957 to propose the central dogma of molecular biology. And I spent a lot of time on his original papers in our stack, because the way we talk about the central dogma today isn't exactly what Crick originally meant.
No, it really isn't.
We usually hear it as DNA makes RNA and RNA makes protein. But Crick was specifically talking about the flow of information, right? He was driving a stake through the heart of that old protein-first theory we just talked about.
He was drawing a very hard line. His core assertion was that sequential information cannot flow backward from a protein to a nucleic acid.
Meaning once it's built, it's built.
Right. Once the information has been translated into a 3D protein structure, that protein can never serve as a template to reconstruct RNA or DNA.
But of course, biology despises a strict rule. And the further I got into the sources, the more I realized almost every modern textbook rule about this process comes with a massive asterisk.
Oh, absolutely.
Take the old idea of one gene, one protein. The math absolutely fails here. Humans only have about 20,000 protein-coding genes, but our cells manufacture well over 100,000 distinct proteins.
The mechanism that breaks that math is alternative splicing.
Which we touched on earlier with the introns.
Right. Discovered in the late 70s by Sharp and Roberts. Going back to those spacer regions in the pre-mRNA, the spliceosome doesn't just cut out the introns, it can also selectively cut out certain exons, leaving them behind.
So it edits the actual coding parts too.
Yeah. Depending on which exons the cell chooses to include or skip, a single gene's transcript can be spliced into dozens of different mature mRNAs, producing entirely different proteins.
So you take one massive chapter of the blueprint, and depending on which paragraphs the editor redacts, the blueprint builds a completely different machine.
It allows immense biological complexity from a relatively compact genome.
Another asterisk I noticed relates to Crick's directional flow of information. The pop science version of the central dogma says information only flows forward, DNA to RNA to protein. But then I hit the research on retroviruses like HIV.
Right, which totally flipped the script for a lot of people.
HIV doesn't have DNA. It carries its genome as RNA, invades a cell, and uses an enzyme called reverse transcriptase to write that RNA backward into the host's DNA.
This is a perfect example of how scientific nuance gets flattened over time, honestly. When reverse transcriptase was discovered in 1970 by Temin and Baltimore, many headlines claimed the central dogma was dead.
But it wasn't.
No, because Crick's actual precise paper never forbade RNA being reverse copied into DNA. He had even anticipated it as a theoretical possibility.
Oh, I didn't realize he predicted that.
Yeah, the only thing he strictly prohibited was information flowing out of a protein.
So the original genius holds up, even if our shorthand for it was wrong. But, there was a plot twist in these papers that genuinely threw me.
Which one?
For decades, the entire scientific community assumed the ribosome was a protein machine that built other proteins. The assumption was that enzymes are proteins, so the catalytic core doing the heavy lifting in the ribosome must be a protein.
Oh, yeah. The paradigm shift around the year 2000 was just seismic for that.
Tell me about it.
When researchers finally crystallized the ribosome and looked at its 3D atomic structure, they realized the proteins are mostly just structural scaffolding on the outside.
Just the shell.
Right. The catalytic core, the actual active site that physically bonds the amino acids together, is made entirely of RNA.
The ribosome isn't an enzyme, it's a ribozyme.
Exactly.
The RNA itself is acting as the chemical machine. That completely reshapes how you think about RNA. It's not just a passive message tape, it's doing the mechanical labor.
It really is a marvel.
And speaking of proteins breaking the rules, the last major exception to the biological textbook involves prions.
Prions are a profound exception, discovered by Stanley Prusiner in the 90s. These are responsible for neurodegenerative conditions like mad cow disease, and they are entirely devoid of DNA or RNA.
None at all.
None. A prion is simply a normal cellular protein that has folded into an aberrant, highly stable, three-dimensional shape.
But the terrifying mechanism here is that it acts like a zombie. It doesn't need a nucleic acid template to reproduce.
The reproduction is purely physical. When a misfolded prion encounters a normal, correctly folded version of that same protein, it physically interacts with it and forces the normal protein to undergo a conformational change.
It literally bends it out of shape.
Yes, misfolding it into the prion shape. It creates a cascading chain reaction of misfolded proteins that eventually destroy the brain tissue. It completely upended the bedrock assumption that a transmissible biological trait always requires a nucleic acid.
Wow. So we have cracked the code, we've watched reverse transcriptase flip the direction, and we've seen RNA act as a machine. But stepping back from the theoretical, the source material makes a hard pivot into the physical stakes of all this.
The real-world impact.
Exactly. Because this microscopic translation process is the singular link between a genotype, the DNA you inherited, and your phenotype, the actual physical reality of your body.
Yeah, the translation machinery builds the actin and myosin that contract your muscles, the enzymes that digest your food, and the hemoglobin that transports your oxygen.
And when we look at a molecular disease like sickle cell anemia, we see exactly how fragile this mechanism can be.
Vernon Ingram's work in 1956 isolated the exact defect, and it is almost unimaginably small. It is a single nucleotide substitution in the gene for beta-globin.
Just one letter.
One. Out of the 146 amino acids that make up this chain, just one codon is mutated from JAG to GTG.
And that single change tells the tRNA to bring the amino acid valine instead of glutamic acid to position 6 on the chain.
Yeah.
And the why of what happens next is purely chemical, right?
It's all chemistry. Glutamic acid is hydrophilic, meaning it loves water.
Oh, wait, yeah.
Valine is hydrophobic, it repels water.
Right.
By placing a hydrophobic spot on the outside of the hemoglobin molecule, the proteins suddenly want to stick to each other to avoid the watery environment of the blood.
So under low oxygen conditions, they clump together into these massive, rigid fibers. That single wrong amino acid physically warps the entire red blood cell into a sickle shape, causing vascular blockages, immense pain, and systemic organ damage.
It's devastating. One wrong read by the ribosome ripples out to alter a massive physiological system.
But the flip side of that fragility is engineerability.
Yeah.
Because the genetic code, the matching of codons to amino acids, is virtually universal across almost all domains of life. We can manipulate the system.
Which is incredible. The tRNA puzzle pieces in a bacterium operate on the exact same geometric rules as the tRNA in your cells.
Which is how Genentech revolutionized diabetes treatment in the early 80s. Prior to that, insulin was harvested from the pancreases of pigs and cows.
A very slow, limited process.
Very. But researchers took the human gene that codes for insulin and spliced it into the DNA of E. coli bacteria. The bacterial RNA polymerase transcribed it, and the bacterial ribosomes translated it.
The bacterial machinery doesn't know or care that the sequence originated in a human. It simply reads the mRNA tape and outputs human insulin.
We turned bacteria into microscopic pharmaceutical factories.
Exactly.
And we scaled that concept up to a global population level with the mRNA COVID-19 vaccines. Reading the breakdown of how these vaccines work completely dispels the persistent myth that they somehow alter your DNA.
Right, going back to our restricted library analogy, it's physically impossible.
Because the blueprint vault is locked.
The mechanism prevents it entirely. The vaccines deliver a lab-synthesized mRNA sequence encoding the viral spike protein. That mRNA is deposited directly into the cytoplasm, the workshop floor.
So it never even sees the nucleus.
Right. It lacks the chemical signals to cross the nuclear membrane, so it can never enter the vault where your DNA is kept. It simply temporarily hijacks your cytoplasmic ribosomes. They read the tape, build the spike protein to train your immune system, and then natural cellular enzymes shred the synthetic mRNA within days.
The blueprint is gone, but the security system is trained.
Exactly.
The last practical application that really stood out in the sources involves antibiotics. If I have a bacterial infection and take something like tetracycline, it has to kill the bacteria without destroying my own cells.
Right. You don't want to stop your own protein synthesis.
No. And that selectivity comes down to the subtle architecture of the ribosomes themselves.
Human ribosomes and bacterial ribosomes diverged evolutionarily a very long time ago. Humans operate with what are called ADS ribosomes, which are slightly larger and structurally distinct from the 70S ribosomes found in bacteria.
So they're different models of the same fundamental workbench.
which allows pharmacological precision. Antibiotics like tetracycline or streptomycin are molecules shaped specifically to wedge into the gears of the bacterial 70S ribosome,
Just jam in the machine.
physically blocking the tRNA from locking into place. It completely shuts down bacterial protein synthesis, starving the bacteria. But the drug physically cannot bind to your human ADS ribosomes. Your cells just keep building proteins as if nothing is happening.
The difference between wiping out an infection and wiping out your own cellular machinery comes down to a microscopic structural gap.
It's all about shape.
Well, we have traced a massive journey through these sources today. We started with the static, locked-away DNA vault, watched the RNA polymerase forge a working copy, saw the spliceosome surgically edit it, and followed it out to the cytoplasm where tRNA puzzle pieces and ribosome machinery physically assemble the structures of life.
It's a lot to take in, but the foundational takeaway here is that grasping the mechanics of transcription and translation is no longer just for biology students.
No, not at all.
It is the absolute bedrock of modern therapeutics. Every future leap in fighting genetic diseases, targeting cancer mutations, or deploying rapid vaccines will rely on intervening at some point along this specific pathway.
It is the ultimate translation of information into physical reality. But, this source material leaves us with one final fascinating implication to chew on. We talked about how the core of the ribosome, the central machine of life, is a ribosome. It's made of RNA.
Right, RNA doing the heavy lifting.
If RNA has the capacity to store genetic information, like a blueprint, and it has the chemical ability to act as a machine that builds things, it solves a massive chicken and egg problem for the origins of life.
It really does.
Does this mean the earliest life forms on Earth didn't need DNA or proteins at all? Could we be the distant descendants of an ancient RNA world where RNA acted as both the architect and the construction worker, doing absolutely everything itself?
It's a profound thought.
It is. Something to think about the next time you look in the mirror and wonder what's really running the machinery inside you. Keep questioning, keep exploring, and we'll catch you on the next deep dive.